Hello everyone and welcome to Botany 101 with Gaurav. Today we are going to start with photosynthesis in higher plants. The term photosynthesis refers to the manufacture or synthesis of food in the form of glucose or starch in plants. In this chapter we are going to cover photosynthesis in higher plants. The word photosynthesis is made up of two words. Photo, that comes from the word photon, which means light, and synthesis, which means to manufacture complex organic food substances. So the term was given by Charles R. Barnes in 1893. So the term photosynthesis was given by Charles Barnes in 1893. So let's move on. They are saying that it is a process in which green parts of the plant means wherever chlorophyll is present, that part of the plant would appear green. It could be the leaf, it could be the stem, and in some extreme cases it could be the root. So they are saying that green parts of the plant synthesize or manufactures complex organic food substances, which is generally glucose stored in the form of starch. Now glucose is a monosaccharide while it is stored in the form of a polysaccharide known as starch. This complete reaction, it utilizes carbon dioxide and water, and it occurs in the presence of sunlight. Carbon dioxide is utilized, water is utilized, sunlight is utilized. This reaction takes place inside chloroplast. What is produced? Glucose is produced, food is produced, and oxygen is produced as a byproduct. So if you look at the reaction, it says carbon dioxide plus water gives you glucose, water, and oxygen as a byproduct. So what kind of a reaction is photosynthesis? One may ask. Photosynthesis is an anabolic reaction. What is an anabolic reaction then? A reaction in which simpler molecules join together to form complex molecules. You can see that carbon dioxide, being a simpler molecule, is being joined carbon by carbon one together to form a larger molecule known as glucose, and glucose, being a monosaccharide, a monomer, is joined again and again repeatedly to form a polysaccharide known as starch. Now you will be amazed to know that the plants manufacture or synthesize the first product of photosynthesis is glucose, which is a monosaccharide. But the plants store glucose in the form of polysaccharide known as starch, but when it comes the time to transport this sugar, they transport it into the form of a disaccharide known as sucrose. So this was interesting enough. Now the next thing is that it is an endergonic process. What do you mean by an endergonic process? Since this process requires energy, the energy is not coming out of the reaction, the energy is getting into the reaction. We need energy so that the reaction moves forward. So in this particular case, this is an endergonic reaction requiring energy in the form of light, usually coming in from the sun. The last thing they are telling you is that this is an oxidoreductive reaction. What do you mean by this is an oxidoreductive reaction? Now just try to understand it very carefully. Whenever we say that a molecule or a species has been reduced, reduction means you are accepting electrons, you are taking electrons. Reducing somebody else means you are giving them electrons. Is that much clear? So if you look at the equation, carbon dioxide here is being reduced to carbohydrate, which is the glucose, whereas water is being oxidized. How is that happening? Well, in biological classification in the second chapter, you have already been through this, that in photoautotrophic organism, you need a compound which is oxidizable, easily oxidizable compound, that is both a proton donor and an electron donor. You remember that, right? So here the splitting of water is going to get split, and there you are going to get electrons and protons, and those protons and electrons are going to reduce your carbon dioxide into a carbohydrate, which is generally glucose. So here you can understand that there is a very important role of sunlight in our life, in our sustenance. You know, in the sustenance of all life on this planet, sunlight plays the major role, because whatever food we eat is coming out of photosynthesis, whatever oxygen we breathe is coming out of photosynthesis, and whatever fossil fuels we are using, that also is coming out of photosynthesis. So I hope you understand how significant this is. Now, what are we going to study in this chapter? Number one, we are going to understand the metabolic machinery of photosynthesis. What are the molecules? What are the pigments involved? Number two, we are going to study the various reactions, light reaction, dark reaction, various phases, means the same thing here. And lastly, we are going to understand what are the factors that affect the rate of photosynthesis. Moving on, they are saying what is the importance of photosynthesis? See, number one, it is the primary source of food on earth. Firstly, the radiant energy of the sun is converted into chemical energy, glucose, stored in the form of starch, broken down again in organisms, and released and transiently stored in a molecule known as ATP. This is the whole story. You already know that light will come from the sun, that light will be trapped by chlorophyll, then that light energy would be used by oxygen evolving complex to break down water and to liberate oxygen. Also, in this particular process, electrons will be released. Those electrons and protons will then form a gradient. Actually, only the protons will form the gradient known as the electrochemical gradient or the electrochemical proton gradient. And later on, we are going to use that electrochemical gradient to generate ATP and NADPH. Subsequently, ATP and NADPH would be used to produce glucose. So this is a cycle where sunlight enters, that light energy is converted into radiant energy, that radiant energy is stored in the form of a bond, in form of bonds in chemical energy form. So now moving ahead, what are the basic requirements or raw material of photosynthesis? Number one, carbon dioxide is required. Number two, water is required. Number three, sunlight is required. And number four, chlorophyll is required. So we are going to go through a list of basic experiments in here to try to understand how do we need these requirements. What are the basic requirements? These are the four basic requirements. How do you came to conclude that these are the requirements? Let's try to find out. So first of all, we are going to figure out how chlorophyll is required for photosynthesis. In this experiment, what we have done is that we have taken a simple plant, a variegated leaf plant. What do you understand by a variegated leaf? A leaf in which we have green regions where chlorophyll is present and non-green regions where chlorophyll is absent. Such leaves are called as variegated leaves. You have already seen these leaves in your home, right? You have seen money plant. That's an example. Another example is in front of you, which is croton. So we take a plant, we de-starch it. How do we de-starch a plant? See, whenever a plant is in the presence of sunlight, it will have that basic raw material fulfillment to do photosynthesis and produce sugar. That sugar, the plant itself, it's going to use that sugar for respiration. So even if a plant is producing sugar, after a certain point of time, if it is not synthesizing any more sugar, the same amount of sugar would be gone. Why? Because the plant itself would be doing respiration and using that energy. That is understandable. Okay. So we have a plant, we are de-starching it by putting it into a dark place out of sunlight for, let us say, 2 to 3 days. It has used up all of its starch. So there is no more starch. So we have it in 3 days in the dark. So the plant is like this. It is de-starched. Next, what am I going to do? I am going to put the same plant now in sunlight. Okay, you did that. What's going to happen? As soon as the plant will get some sunlight, it will start doing photosynthesis and it will start producing glucose once again. And then what we are going to do, we are going to test the region of both the green and non-green regions for starch. And when we did the test, we figured out that the green part turns blue-black, test positive for starch, whereas the yellow part, non-green part does not turns blue-black. That means test negative for your starch. Why is it happening? Because chlorophyll is required for photosynthesis. In the non-green part where chlorophyll is absent, there is no photosynthesis happening. Okay. Now, what is the other experiment? The other experiment is to determine in the same way whether light is required or not. So we are going to do the same experiment. We are going to de-starch the plant, but this time we are going to put a paper clip, black color paper clip upon the leaf so that no light can enter this particular region. Now we repeat the same experiment again, and when we check this particular leaf for starch, so what we are going to find out, what is going to be the conclusion? That the green part will turn blue-black, testing positive for starch, whereas the black part, the darkly shaded part which was out of the light, will not have any light, and therefore no photosynthesis is going to happen here. So we have established two facts. First, chlorophyll is required, and second, light is required. Now there is one another way by which we can do this chlorophyll experiment. The other way is that if you don't have access to a variegated plant, you can do the same experiment by using alcohol. So you wash a part of you alcohol wash the a particular part of the leaf, and the chlorophyll from that particular part would be dissolved away. Now comes the question, what does alcohol wash do to the chlorophyll? The answer is alcohol washes chlorophyll out of the leaves by breaking down the chlorophyll and dissolving it into the alcohol. So this is the first thing that's going to happen. So sir, you have been talking about starch test, starch test, starch test. What exactly is starch test? So how does starch test works? The basis of the starch test is the reaction. Reaction between what? Reaction between iodine and starch. When iodine solution, usually iodine dissolved in potassium iodide, comes into contact with starch, it produces a blue-black color. Okay, this reaction occurs because iodine molecules fit inside the helical structure of amylose. So this reaction is not going to happen with amylopectin, but with amylose. Now moving ahead. Now next experiment is to determine whether or not carbon dioxide is required. Now this next experiment is simply known as Moll's half-leaf experiment. I will tell you a trick to remember these experiments. The first experiment was to determine whether chlorophyll was required. That experiment is known as alcohol wash experiment or variegated leaf experiment. The second experiment was to determine light, and that was black paper experiment. And the third is to determine your carbon dioxide, which is known as Moll's half-leaf experiment. It is a very simple experiment. Same way we are going to destarch the plant, and then again we are going to put it into sunlight. But this time what we are going to do is that we are going to put half the part of the leaf, half of the leaf, inside a container which is containing KOH, potassium hydroxide. Since we all know that potassium hydroxide absorbs carbon dioxide, there will be no carbon dioxide in this part of the experiment. Therefore, this part of the leaf will not be able to do photosynthesis because of the unavailability of carbon dioxide. So this time when we are going to do starch test, half of the leaf that had access to carbon dioxide will give us blue-black color, whereas half of the leaf which does not have access to your carbon dioxide will not give us blue-black color. So this was your Moll's half-leaf experiment. Now coming to early contributions in photosynthesis, the first person that comes to our mind is Joseph Priestley. The second person is Jan Ingenhousz. The third person is Julius von Sachs, then Engelmann, and then Van Niels, and then at the last Reuben, Rendell, Kamen, and Hyde. Let us see one by one what are these experiments and what are the contributions. So the first person is Joseph Priestley. Now this guy said that there is an important role of air in the growth of green plants. Number one. Second, he discovered oxygen in the year 1774. And lastly, he did a very basic experiment, very nice experiment. What he did that, he took a container, you know, a bell jar, and you take a mouse and you put a bell jar over a mouse. What will happen? After some time, the mouse will die because of the absence of oxygen. Similarly, if you have a candle and you put a bell jar just over it, right? What will happen? After some time, the candle will extinguish because there will be no oxygen. Now in the third condition, what this guy did is that he put a bell jar just over, you know, a mice and a candle, but this time he also put mint plant inside. So what will happen? Mint plant will produce oxygen by photosynthesis and will consume the carbon dioxide given out by mice and the candle. So this is how he explained that something out of the air was taken by mice and candle. Something out of the air was taken by mice and candle, but was returned to air by the mint plant. So there is a cycle. Something was taken out by the mice and candle, and the same thing was returned by the mint plant. Let us see. So this was the basic experiment. He put mice and candle, both of them die. He put mice and candle with the mint plant, both of them, all of them survive. So once again, 1770 revealed the role of air in the growth of green plants, discovered oxygen, and then did the basic experiment we were talking about. So our next scientist is Jan Ingenhouse. Now this guy, he said that, okay, you are saying that oxygen is required or air is required for the growth of green plants and for photosynthesis. Nice and fine. Secondly, listen to me. Other than that, sunlight is required, and he proved this through an experiment. He took this aquatic plant hydrilla, and he placed this hydrilla into that same experimental setup. Now what will happen? In the green parts of the plant, wherever the sunlight can reach, we will see bubble formation, and the bubbles will be formed of oxygen. So Jan Ingenhouse, through this basic experiment, has identified that these bubbles were indeed made up of oxygen, and you will only find bubble formation when light availability is there. If you place this experimental setup in the dark, there will be no bubble formation, but in the light, bubble formation would be there. Now after Jan Ingenhouse, our next scientist is Julius von Sachs. Now Julius von Sachs, what did this guy said? Very simple things. First, whenever there is a growth in green plants, you will find glucose over there. First thing. Second, glucose is the product of photosynthesis and is stored as starch. Second point. Very, very important point. So he said that green plants is where glucose is formed, and glucose is usually stored as starch. Next important thing, he said that the green colored pigment that you are finding, you will find it in special bodies in plants, and those special bodies, now we know that those special bodies are known as chloroplast. You know that chloroplast was discovered by Hugo von Mohl. Okay. Now our next scientist is T.W. Engelmann. According to me, out of all the photosynthesis experiments, this is the best experiment that shows us that with minimal resources we can do a lot more than, it's not necessary that we need a lot of resources to do a lot of things. Even with minimal setup, minimal resources, if we put our mind to maximum efficiency and usage, we can draw better conclusions. Let us try to understand what did this guy do. Okay, let us just try to understand. This guy did a very basic experiment. So you have cladophora. Cladophora is a green algae, and you have a suspension of bacteria, of aerobic bacteria. So what is the setup? What does the setup looks like? Just try to understand. This is my petri dish. I am having aerobic bacteria suspension in that. So everywhere there is aerobic bacteria. Let's just try to mark it out. Everywhere there is aerobic bacteria. Other than that, what this guy did is that he put a prism in here. If white light is entering here, it would be split into its spectral components like this. If I'll start writing down the components, it would be Webb gear. So let us say this is the red component here, and here is the blue component. Now this guy was a genius. How? Is by doing this simple experiment, putting cladophora, the green alga, in this setup, he observed that the growth of aerobic bacteria. Where will the aerobic bacteria grow? Only where oxygen is present. Where will oxygen be present? Only where photosynthesis will happen. Because cladophora will do photosynthesis, it will release oxygen. That oxygen would be used by the aerobic bacteria. So the growth of aerobic bacteria in this particular setup would only be at the place of photosynthesis, and Engelmann figured it out that it was only the red region and only the blue region where there was growth of aerobic bacteria. So this guy, even without doing any spectrophotometric studies, absorption, this, that, anything, he said that blue and red are the regions of light where maximum photosynthesis occurs. Now this is the active spectrum of your photosynthesis. Now this guy, without doing any complex you can say experiments, draw a very significant conclusion. So this basic equation was given by Engelmann. This says that carbon dioxide reacts with water to form carbohydrates, sugar. The next scientist is Cornelius van Niel. Guy is a microbiologist, and he studies purple and green sulfur bacteria. You know from biological classification that other than water, any other oxidizable compound can also act as a proton and electron donor, right? So this guy said that it is not necessary that the oxygen that is coming out of photosynthesis, it's not coming out of the breakdown of carbon dioxide. When I saw the equation for the first time myself, carbon dioxide plus water gives carbohydrate, it was pretty clear to me that carbon dioxide was going to break down into oxygen. So what did I thought? That oxygen coming out of the end is actually coming out of the breakdown of carbon dioxide. But this guy, he said that no, 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 no. You will have to figure it out yourself that it can be any oxidizable compound. If in that equation it is an H2A, then we are going to get an A. See what's happening? Just try to understand. Carbon dioxide is reacting with a highly oxidizable compound H2A, which is going to denote proton, right? And those protons are going to be used up into the formation of this sugar. And whatever it is carrying, if it is an A would be gone here. Now, if in this same equation I have carbon dioxide plus H2O, then H2O will split. How? Let us just try to understand. H2O will split into 4 protons plus 4 electrons plus a molecule of oxygen. And these 4 protons are actually being used to combine with this carbon dioxide to produce this sugar. In the same case, if water is being replaced by hydrogen sulfide, now protons are going to be gone here and sulfur will be coming out of the end. So this important contribution is coming out of Cornelius van Niel. But this was confirmed by radioisotopic studies by Reuben, Randall, Kamen and Hyde. They used non-isotopic, non-radioactive stable isotope of oxygen 18O to prove that oxygen coming out of the photosynthesis is actually coming out of due to the splitting of water. It's coming out of water and not out of your breaking down of carbon dioxide. So guys, let's move ahead. So in early contributions, there is one more important contribution by Robert Hill. See, photosynthesis comprises of 2 parts. We have already discussed that: light reaction and dark reaction. Light reaction is also known as Hill's reaction, whereas dark reaction is also known as Blackman's reaction. So Robert Hill is the scientist upon whose name this Hill's reaction is being given. So what did this guy do? He used electron acceptors other than carbon dioxide. Which are these electron acceptors? DCP-IP, that is dichlorophenol, indophenol, ferricyanide, chromates and benzoquinones. The ferricyanide is reduced to ferrocyanide. He was able to demonstrate evolution of oxygen even in the absence of carbon dioxide. So this was one of the major contributions by this guy. Other than this, there are a few scientists whose contribution is also important and may come in the exams. It is very easy. Let's try to understand. John Senebier, who is a Swiss scientist, he said that simply that fixed air, that carbon dioxide was required for photosynthesis and it was absorbed from the air and combustion supporting oxygen was released. 3 things he said. First, carbon dioxide is being used. Where is it coming from? Coming from the atmosphere. And lastly, oxygen is being evolved. Next, Nicolas Theodor de Saussure, a Swiss scientist, he said that plants cannot survive in the absence of they cannot survive in the absence of oxygen and carbon dioxide both. They need both oxygen and carbon dioxide. The volume of carbon dioxide used is equal to the volume of oxygen evolved in photosynthesis. But he said that this oxygen was derived from carbon dioxide. This is wrong. This oxygen is coming from the breaking down of water or splitting of water. He also proved that water is important for photosynthesis. Other than that, Pelletier and Caventou, they are 2 French scientists. Their important contribution is that they said that chlorophyll, which is a green pigment, you can extract it out by alcohol wash. We have already understood what is the chemistry behind it. Lastly, Hugo von Mohl discovered chloroplast, and Julius Mayer, he said that the sunlight is being converted into chemical energy in photosynthesis. Now next important question is, where does photosynthesis takes place? Now listen very carefully. Photosynthesis takes place in green parts of plant. Now green parts of a plant could be, it could be a leaf, very nice. Other than the leaf, it could be a stem, it could be a flower, it could be a fruit, it could be a root. We are going to look at those structures too. Okay, fine. You are in a hurry. Let's look. So can you guess example where non-leafy structures, they are doing photosynthesis? We have studied them in morphology of flowering plants. Number one, roots. Assimilatory roots which do photosynthesis are found in Trappa and your Tinospora. Phylloclad, which is a flattened stem able to do photosynthesis, is found in Opuntia, Euphorbia, and petiole, flattened petiole that can do photosynthesis, is found in your Acacia. Lastly, young fruits, they are also able to do photosynthesis, as in the case of apple and avocado. So if I am talking about leaf, can you draw the structure or diagram of a leaf and tell me exactly where photosynthesis is happening? Let's try to figure it out. So I have a very neat and simple trick of, drawing this particular diagram. You don't have to do much. A straight line here, almost a straight line here, almost a straight line here, leave a little bit gap and go like this, a little bit ahead. Okay, this is the first thing to be done. Next, what you do is that you draw two kidney beans. This is your stomata. Other than that, you will draw a very fine line, because these flattened cells, thin flattened cells are actually your epidermis cell. So guys, similarly, we are going to draw one more epidermis here. So students, which is which? So this one, I'm going to draw an additional layer over this one, and let's do this to show that this is separate. So an additional layer over the leaf can be found in some xerophytic plants, which is generally made up of cutin, and this layer is known as cuticle. Beneath the cuticle, generally in the case of leaves, is your upper epidermis. This is your lower epidermis. This is your stomata. Stomata, guys, is the opening in leaves that helps in the gaseous exchange. Now, very simple and neat trick. You will have to draw these vertically long cells simply like this, and inside these are the chloroplasts. Can you tell me which layer is this? Yes sir, this is mesophyll. Which mesophyll is this? This is palisade mesophyll, and beneath palisade mesophyll there is an additional layer, and this layer is your spongy mesophyll. So I'll draw it just like this. For ease, I'll draw it like this. So this is your spongy mesophyll. I have asked you, where does photosynthesis takes place? Green parts of the plant. Let us consider leaf. Inside leaf, where is it going to happen? In palisade mesophyll, let us say for example. Inside palisade mesophyll cells, where exactly is photosynthesis going to happen? Let's try to understand. What is inside a cell in which photosynthesis takes place? Which is structure? It is chloroplast. Chloroplast is the cell organelle inside which photosynthesis will take place. So looking at the structure of a typical chloroplast, this is the outer membrane, this is the inner membrane, and this is the intermembrane space. Chloroplast and mitochondria are semi-autonomous organelles. They have their own protein synthesizing machinery, their own ribosomes, their own DNA. So they will have their own DNA, they will have their own ribosomes. The ribosomes will be 70S in nature. There will be stacks of thylakoid. Thylakoids are membrane bound structures, right? Thylakoid stacks will be known as granum. Single stack is granum, multiple stacks are grana. The two stacks or different stacks are connected by a lamellae, which is known as stroma lamellae, and all the interior space is known as the stroma. The light reactions and the dark reactions, we are going to find out soon, occur separately. We are going to find out soon. So we have already discussed this. What is your chloroplast? We know we have already studied the structure of chloroplast. Do you wish to draw me a structure of chloroplast? Well, let's try to do. So guys, if you want to do a chloroplast, very simple. Chloroplast is a double membrane bound organelle. So you don't have to worry much about it. Just draw a double membrane bound organelle. So this is your outer membrane, inner membrane and intermembrane space. First of all, we are going to draw this. This is a single thylakoid. So we are going to draw a little bit more like this. Now this is stack. Single stack is known as granum. Now I am going to draw another stack. And connect these two. Let's try to label. This is your outer membrane. This one is your inner membrane. This is your intermembrane space. This is your thylakoid. This is your singular is your granum, and these two together are grana. This is a stroma lamellae. Stroma lamellae. Now, when photosynthesis will happen, what will happen? We will have synthesis of starch granules. So these are starch granules. We'll have some DNA. We'll have ribosomes. And we have stroma. So this is a typical structure of a chloroplast, right? Now moving ahead, this is what we are going to talk about now, that in a single chloroplast we are talking about, there is a division of labor. Light reaction and dark reaction occur separately. Let's try to understand what they are. When we are talking about photosynthesis, the first thing that is going to happen is the light reaction. Light reaction mein what will happen in light reaction? The first thing that is going to happen in light reaction is the absorbance of light. First of all, light will be absorbed. Second, using that energy, water will be split. So the second stage would be the splitting of water. The third stage will be to produce, intermediates, energy intermediates known as NADPH and ATP. So light reaction is this much. You will absorb light, you will use that light energy to split water, you will use those proton to generate an electrochemical gradient or a proton gradient, and then you will use that proton gradient to generate ATP and NADPH. So this part of the reaction known as the light reaction occurs specifically if this is a thylakoid, then the enzymes for light reaction are embedded into the thylakoid membrane. This is thylakoid. If I'll draw this another external membrane layer, this is the chloroplast. So inside the thylakoid, this is the thylakoid. Inside the thylakoid membrane, enzymes for light reaction are there, and inside the stroma, the enzymes for dark reaction are there. What is dark reaction? Once ATP and NADPH have been formed, then we will use those ATP and NADPH to fix carbon dioxide and produce glucose. That is where C3 cycle and our dark phase comes into play. Moving ahead little by little. Before moving ahead, let's try to understand what is light. Light is an electromagnetic wave. It is a part of the greater electromagnetic spectrum. The electromagnetic spectrum has the highest energy and shortest wavelength, gamma rays. Then moving towards radio waves, they have the lowest energy and longest wavelength. And in between infrared and ultraviolet, we have the visible light region. So if we are talking about the visible light region, this is it. It starts from 400 to 700 nanometers, and specifically photosynthetically active radiation is the radiation that is falling in 400 nanometer to 700 nanometer range. So the light in this particular range is known as photosynthetically active radiation because it is or can be utilized for the synthesis of food by the process of photosynthesis. So what we can see here is that red and blue, these are the two important regions where photosynthesis occurs as predicted by Engelmann. Now moving ahead, how is going photosynthesis going to occur? First of all, light is going to be absorbed. Who is going to absorb that light? Photosynthetic pigments are the substances or the molecules that are going to absorb light. Number one, a chromatographic separation. If we do a chromatographic separation of a leaf, we are going to find out four different kinds of molecules. Which are these? Chlorophyll A, chlorophyll B, carotenoids and xanthophylls. If we talk about chlorophyll A will appear blue or bright, will appear blue green. Chlorophyll A will appear blue green, whereas chlorophyll B will appear yellow green. Now xanthophylls will appear yellow, whereas carotene will appear yellow to yellow green. So see, carotenoids are the bigger group. Inside the carotenoids are the xanthophylls and carotenes. Okay. Now chlorophyll A is the primary photosynthetic pigments, as we are going to figure out later that it is the reaction center. What is the reaction center? We'll see, just wait. So what is the structure of chlorophyll like? If somebody asks you what is the structure of chlorophyll like, so you will say that chlorophyll has a tadpole like structure. What is a tadpole like structure? It has, they say, a porphyrin head, and that porphyrin head is attached to a phytol tail. So this porphyrin head is polar in nature and this phytol tail is non-polar in nature. So this is the layer, this is the part of the molecule which is embedded inside the lipid bilayer. You already know that the lipid bilayer is non-polar in nature or hydrophobic in nature. So there must be some hydrophobic part that is going to attach to it. This is the hydrophobic part, whereas the part which is polar, which is outside the membrane, this is the polar part. So let's look at the diagram. This is the porphyrin head. It contains 4 rings. This is otherwise known as a tetrapyrrolic ring structure, tetrapyrrolic cyclic structure containing 4 pyrrolic rings, and there is a phytol tail. They say that chlorophylls have a complex ring structure chemically related to the porphyrin-like groups found in hemoglobin and cytochromes. It is the site of electron rearrangement. We are just going to figure it out. It contains cyclic tetrapyrrolic structure with non-ionic magnesium. You will have to remember that at the center of this structure, at the porphyrin head, is a magnesium, okay, non-ionic, and it is attached to 4 nitrogens. If you remember this much, you are going to do well in your exam. Now the phytol tail is a long hydrocarbon tail, almost always attached to the ring, okay, and anchors the chlorophyll to the hydrophobic portion. So this is the part which is going to be embedded into the thylakoid membrane. They say that there are different types of chlorophyll, chlorophyll A, B, C, D and E, and the chlorophylls that are found in bacteria, bacteriochlorophyll A and B. So what is the difference between chlorophyll A and B? See, here you only have this CH3 methyl group. If on ring 2 I replace methyl group with a formyl group, then this is going to become chlorophyll B. Okay, so they say that in chlorophyll B, formyl group replaces methyl group on ring 2nd. So what are accessory pigment? Just try to understand it very simply. I'll try to explain it with the help of a diagram. It's very, very simple. I have a molecule over here. Light is falling in the form of photons. Not a, just not a single continuous ray, but different packets that are going to reach. Let us assume for an instance that at time t is equals to 1, this particular molecule, which is the chlorophyll A molecule, it absorbs a particular photon and it emits an electron or excites an electron, or the electron of chlorophyll A reaches into a higher energetic state, which is the chlorophyll A star. Now this electron, this has moved to a higher energy state, so there is a vacancy over here. Now if another photon strikes, this vacancy cannot be, another electron just cannot go because there is already a vacancy over there. First thing. So this vacancy needs to be fulfilled. The electron that has moved from the reaction center, chlorophyll A, you need to fulfill this vacancy. This is fulfilled by a particular complex known as oxygen evolving complex. This oxygen evolving complex has 4 manganese ions, one calcium and 1 chloride ion in it. Okay, and what is the function of this OAC? It splits water. It uses light energy to split water into 4 protons, 4 electrons and oxygen. And these electrons now would be sent here to fulfill this vacancy, so that this cycle goes on continuously. But okay, now one problem is solved. Continuously you will be getting electrons by splitting of water. So they said that light of different wavelengths are absorbed by these antenna molecules, because chlorophyll A has a particular absorbance. So other than that absorbance, for that wavelength, these molecules, which are antenna molecules, they will be absorbing the light, and they will then transfer the energy to the reaction center via electron spin resonance. Okay, they also protect the chlorophyll A from excessive heat and photooxidation, and therefore are known as shield pigments. So guys, moving ahead now. Now carotenoids, they say they are the accessory pigments from yellow to yellow-orange in color, and they absorb visible light mainly in the region of 460 to 550 nanometer. They are hydrophobic, present in almost all parts of the green plants and photosynthetic bacteria. They function to absorb and provide light energy to chlorophyll molecules and prevent them from damage from photooxidation. Now, what is an absorption spectrum? Let's try to understand it. This is a very important concept. What am I going to tell you? So you have a cuvette. What is a cuvette? Cuvette is a glass, imagine a glass tube having water. So this is a glass tube, this has water, and I am throwing light from here. First of all, I send light of 400 nanometer. It passes through, there is a screen. I don't get anything because the molecule that I am dropped in here, it does not absorb light at 400 nanometer. Okay, so simple experiment. I have a molecule A here. This molecule A absorbs light only at 460 nanometer. So when I will send 400 nanometer, no absorbance will be there. I will get this light here. When I will send 401 nanometer, I will get this light here. 402, I will get here. 403 nanometer, I will get it here, because they will pass. They will pass because this molecule A will not absorb them. But similarly, when I will send 460 nanometer, this light will be absorbed, and therefore I will not see this light over here. So what is the absorption spectra? If I send 400 nanometer to 700 nanometer, all the wavelength, then a particular wavelength of light will be missing on the screen, and that is the absorbance. Okay, now this is simple. Let us say, for example, this is where I'll plot my wavelength. Okay, 401 nanometer, 402, 403, 404 similarly. And here is the absorbance of glucose. Here is the absorbance of glucose at which it will absorb. So let us have an imaginary scenario that when I sent 401 nanometer light, it did not absorb, but at 402 it absorbed, and then at 403 is not absorbing, and then it's 0. So we can say that glucose absorbs light of 402 nanometer, but 401, 403, 404 and so on, it will not absorb. Similarly, if we are going to picture it, this is known as absorption spectrum. So what is an absorption spectrum? It is a curve, it's a graphic curve showing amount of energy absorbed at a particular wavelength. So let's try to understand. This is 400 to 700 nanometer wavelength. Here we have given wavelength, here we have given absorption. If I look at the green line, chlorophyll A here is having its absorption somewhere around here. Okay, so if I am talking about 453 nanometer, chlorophyll A will have absorption here, maximum absorption, and here somewhere around 662. Chlorophyll B at here 453 and 642. But if you look at carotenoids, after this particular point, chlorophyll A is not absorbing any light. See, after this particular point, chlorophyll A is not absorbing any light. After this particular point, chlorophyll B is not absorbing any light. But still, for this range, carotenoids are absorbing. So this is the function of antenna molecules. They trap light, they catch light that comes in the range that is outside the collective absorbance range of chlorophyll A and chlorophyll B. What is action spectrum? Now if I try to visualize the rate of photosynthesis on y-axis and the wavelength on x-axis. This particular graph would be known as action spectrum. But sir, how do you, how do you calculate or how do you approximate photosynthesis rate? How do you know how much photosynthesis is going on? Whenever photosynthesis will happen, oxygen will be released. So we take indirectly. So how do we do it? We take an indirect measure by measuring oxygen release. So what is it? If we measure oxygen release over here and we put wavelength in here, then whatever graph we are getting, this particular graph will be called as action spectrum. Now if I will superimpose this graph over this, then it is clear that these two peaks correspond to the peaks of chlorophyll A. So the major portion of this graph action spectrum is coming from chlorophyll A, but still chlorophyll A is not showing any absorbance in this region, but I am getting action spectrum rate of photosynthesis in here, which means photosynthesis is happening in this range and is happening due to either chlorophyll B or due to carotenoids. Okay, so what are they saying? Most of the photosynthesis takes place in the blue and red regions, some takes place in other wavelength, maximum takes place in the wavelength of at the peaks of chlorophyll A. Okay, what are the peaks of chlorophyll A? It has a chlorophyll A has a peak in the blue region in 430 nanometer, whereas chlorophyll B has a peak at 453 nanometer. In red region, chlorophyll A has absorbance at 662 nanometer, whereas chlorophyll B has an absorbance at 642 nanometer. Guys, now we already know about light reaction. Light reaction means absorbance of light, splitting of water, using that electron and protons to generate a electrochemical gradient or proton gradient, and using that proton gradient to generate energy intermediates ATP and NADPH. So in order to do this, first of all we need to trap light, and the smallest unit, the simplest unit that is going to do that for us is the photosynthetic unit or photosystem. So a photosystem is nothing else more than a complex of molecules or different molecules. The core molecule would be the reaction center or chlorophyll A. The chlorophyll A would be surrounded by antenna molecules, and these together would be called as light harvesting complexes. So light harvesting complexes plus reaction center would be called as photosystem. Let's look. So you have a photosystem. It is made up of two things. First is reaction center. There are two reaction centers. Chlorophyll A in PS1 absorbs at 700 nanometer. So this PS1 is known as P700. Whereas the chlorophyll A in PS2 absorbs at 680 nanometers and is known as P680. Other than the reaction center, there are the antenna molecules which comprises light harvesting complex, which is made up of light harvesting pigment and proteins. The proteins, they help to increase the efficiency of photosynthesis. So this is a very basic image. The light is falling. This is the reaction center. The reaction center's electron is getting excited to a higher energy or moving to a next primary acceptor, and the energy is also being, sunlight's energy is also being collected and passed on to the reaction center, right? So we have two photosystems, photosystem 1 and 2, P700 and 680. Let us try to understand what are the differences. P700 reaction center is known as P700, whereas this one is known as P680. PS1 lies on the outer surface of the thylakoid. We will just discuss where the location would be. And these are found in both grana and stromal lamellae. They are found only in granule lamellae. They are involved in cyclic as well as non-cyclic. They are involved in only cyclic, and these are not associated with splitting of water. They are associated with splitting of water. We are going to figure it out just in a moment. So Arnon used the term assimilatory powers for ATP and NADPH. So ATP and NADPH will be formed as an intermediate of light reaction, and these are termed as assimilatory powers. And this particular process of reduction of NADP into NADPH may be denoted as electron transport system, in which electrons will move in a downward hill gradient from one electron acceptor to another electron acceptor placed on a redox potential scale, generating energy in each step, which is used to synthesize ATP and NADPH. This flow of electrons through ATS is also linked to photophosphorylation. We have already discussed what ETS is. ETS or ETC is a series of electron carriers over which electron will pass in a downward journey, and in each step energy would be used to generate electrochemical gradient or proton gradient, and that would be later used to generate ATP and NADPH. Redox potential is nothing but a tendency of a chemical species to get reduced, also known as oxidation reduction potential. Let's try to brief it up very simply. What is happening? Just try to understand. First of all, light is falling on your photosystem 1. Photosystem 2. Photosystem 2, light is falling P680, and the electron is excited to a higher energy state known as P680 star. This vacancy will be filled by splitting of water, and this electron will move to fill this vacancy. Now this electron will move over a series of electron carriers. First of all, pheophytin, then plastosquinone, which will be converted into plastoquinol. Now this is very important. When plastoquinone is converted into plastoquinol, it accepts protons from the stroma. First step. Before this, there was splitting of water, which was already adding protons inside the thylakoid, because these molecules, these photosystems, are embedded in the thylakoid membrane, and on the inner side of the thylakoid membrane is the oxygen evolving complex. So when oxygen evolving complex will break down water, these 4 protons will be accumulated inside the thylakoid. In the second step, when pheophytin will transfer the electron to plastoquinone, plastoquinone will accept protons from the stroma and convert into plastoquinol, and plastoquinol will then transfer the electrons to cytochrome B6, but at the same time will throw the protons inside the thylakoid. So there is an accumulation of protons inside the thylakoid membrane because of 2 reasons. First, splitting of water. Second, plastoquinone takes protons from the outside and converts into plastoquinol. Then it will transfer the electrons to cytochrome B6F, which will transfer it to plastocyanin. Plastocyanin will transfer it to PS1, right? So PS1 is P700, and then PS1 electron at the same time simultaneously was ejected out and converted into P700 star. This electron will now move to ferredoxin, and then to ferredoxin NADP reductase, also known as FNR. So if you can see, this is a non-cyclic photophosphorylation, whereas in another scheme only one part of this scenario works. Let us see. Only PS1 is active. Light photon is falling on your reaction center. The reaction center P680, P700 electrons moves to a higher state P700 star. Now this P700 star electron, rather than moving on towards the right hand side to ferredoxin, it's coming left to via ferredoxin to cytochrome B6, and to cytochrome B6 it will go to plastoquinone, then plastoquinol to cytochrome F, then to plastocyanin, and then it will come back again to its source. So this will be a cyclic one. So this is cyclic photophosphorylation, whereas this is non-cyclic photophosphorylation. So you can see that in non-cyclic photophosphorylation, both PS1 as well as PS2 are used. PS2 comes first, PS1 comes second. But in cyclic photophosphorylation, only PS1 is engaged, PS2 is not involved. So these are the differences. Now if we look at the movement, if we look at the movement of electrons over the series of mobile carriers, what am I going to observe? I am going to observe a Z. If I move it from here to here to here. So this complete scheme, if I'll put all the species on a redox potential scale, then this particular scheme is known as Z scheme. The whole scheme of transfer of electrons starting from PS2 uphill to the acceptor, down to electron transport chain to the PS1, then again to the higher state, and then to NADP reductase. This complete scheme is known as Z scheme. Hill and Bendel suggested the Z scheme of photosynthesis in 1960. Now coming towards our last part in light reaction, this is chemoosmotic hypothesis, which says that for the generation of ATP and NADPH, what do you require? You require a membrane. Let's try to draw this. So this one is very easy. Let's try to link everything now. So this is my thylakoid membrane. This is my thylakoid membrane, and this is my PS2. This is my cytochrome B6F, and this is my PS1. Here I have NADP reductase enzyme. Ferredoxin NADP reductase enzyme is there. Okay. And here I have your ATP synthase. Here you have your ATP synthase. So what is going to happen? First of all, light will fall at the reaction center of PS2. Electron will get excited and move to a higher energy state, and this vacancy will be fulfilled by oxygen evolving complex by breaking down of water. So water will be broken down here, 4H plus 4 electrons plus O2. Oxygen will be evolved. Now this electron, it will move to pheophytin. After pheophytin, it will move to plastoquinone, which will take protons from the outside and convert into plastoquinol, but it will come back again into plastoquinone when it will throw protons in here, right? The electrons will be then transferred to cytochrome B6, which will transfer it to F, which will then will be taken by a mobile electron carrier. Now this you will have to remember, plastocyanin is the mobile electron carrier which will take these electrons to PS1. Simultaneously, light will fall at the reaction center of PS1. This will reach to a higher state of P700 star. This will reach to P680 star. This will move to ferredoxin NADPS reductase, and this particular will take NADP plus H plus from stroma and convert it into NADPH. So by three probable methods, proton gradient is being created. Let us see how a proton gradient is being created. First of all, by the splitting of water, protons are being added into the thylakoid. Secondly, these protons are being removed from the stroma by the plastoquinone. And thirdly, when NADP is converted into NADPH, it is taking protons from the outside, which is the stroma. So by these processes, there is a proton gradient that has been created. More protons are inside and less protons are outside. So ATP synthase, which is an enzyme, it will use this particular proton gradient in a way that it will throw protons out again, and in doing so it will convert ADP plus Pi into ATP. So what are the products of light reaction? The products of light reaction are ATP and NADPH. If only cyclic photophosphorylation will happen, only this part will happen, then only NADPH will form. But if non-cyclic photophosphorylation is happening, then ATP and NADPH both will be produced. So these the products of light reaction, ATP and NADPH, will then be used later in dark reaction in the stroma in order to synthesize glucose using C3 cycle, which was given by Calvin, also known as Calvin cycle, right? I hope you guys enjoyed this much. If you are interested in knowing more, if you are interested in me taking another lecture and explaining to you dark reaction, do let me know.